Hi, my name is Jeremy Arbesfeld, and I am a graduate student in the Wagner Lab at The Ohio State University and Nationwide Children's Hospital. Today, I'll be presenting on FUSOR and the VICC Fusion Curation Interface, a collection of tools that allow for the standardized representation of gene fusions in both clinical reports and genomic knowledge bases. To provide some background, this work has emerged as a collaboration between experts from the Variant Interpretation for Cancer Consortium, or VICC, the Cancer Genomics Consortium, CGC, and the CAP ISMB Joint Cytogenetics Committee. Through biweekly meetings, the group developed a consensus, unified framework for the description of fusion events, paving the way for the development of a set of associated computational tools that allow for this framework to be realized in both clinical practice and genomics research. Here you can see a figure describing features underlying the FUSOR class, a novel data structure whose features were informed by the framework that I previously described. In the interest of time, I won't go over the figure in great detail, but an important takeaway to consider is that FUSOR allows for the representation of both assayed fusions and categorical fusions as well. For those who are unfamiliar with the concept of categorical fusions, this term essentially describes fusions with shared characteristics, such as end-track fusions. By covering both classes of fusions, the FUSOR class allows for fusions to be represented both in clinical and research-based contexts, respectively. Additionally, FUSOR covers both the structural and regulatory elements that comprise fusion events, as you can see on the slide. These structural elements include the gene symbol and associated HGNC ID, transcript ID, and data describing a linker sequence, if present. The regulatory elements, in the context of categorical fusions, include the class, such as the functional domain, the ID, and genomic location. Although the use case of certain elements is specific to the type of fusion that is being understood, FUSOR is again able to model a wide range of fusion events. Here is an example of an EWSR1 assayed fusion with an unknown partner represented using the FUSOR class. As you can see, in describing fusion partners, FUSOR resolves ambiguous gene aliases through normalization and records other relevant metadata, such as the causative event and information regarding the molecular assay that provide important context needed for accurate clinical interpretation. FUSOR objects can be created by supplying the relevant data to classes within the wider FUSOR class. For example, if a tumor sample is sequenced and then processed through a fusion detection pipeline, outputted data can include identification of the involved fusion partners, chromosome number, strands, and breakpoints. Then, this data could be supplied to structural elements of the assayed fusion class, which is a method within the broader FUSOR class, allowing for the fusion event to be described and better understood. For the image on the slide, I should note this is only an individual example of a FUSOR use case, and that more examples can be found in the project's GitHub repository. Here is an example use case for the VICC Fusion Curation Interface, depicting the representation of a TPM3 and Track1 fusion event. This interface leverages the FUSOR class, allowing users to convert fusion event observations to a format that is consistent with the framework that I previously described on an earlier slide, as you can see with the arrows below. Users can then follow the various tabs on the top to add features as necessary, and the Summary tab reports the fusion in accordance with the guidelines that I previously described. This output can also be converted to a JSON, machine-readable format, allowing for information describing the fusion to be directly accessed by the user. Having introduced FUSOR in the VICC Fusion Curation Interface, I would like to briefly mention some additional work that we have planned for the future. First, as this is an open source, community-centered effort, we plan to refine these tools to reflect community needs and feedback through our biweekly meetings. At Nationwide Children's Hospital, we plan to integrate FUSOR into our own fusion detection pipeline to promote more efficient and accurate clinical interpretation of fusion events. Lastly, we plan to integrate FUSOR into knowledge bases such as MetaKB to allow for a better understanding of fusion classes with shared characteristics. This concludes my presentation. I'd like to thank everyone in the Wagner Lab, Nationwide Children's Hospital, and the VICC Gene Fusion Group for their efforts and contributions, and I'm happy to take any questions. Thank you again for your time.